Welcome back to
Welcome
the
back.
first
It's been
official
way too
episode.
long.
It has
Way too long
This is the first official Bio to Bedside episode this year.
of the year.
Yeah, and it's February. So we've taken a bit of a break. We will be back with regular scheduled programming going into the future.
Don't promise too much, Ben.
Yeah. Excellent. All right, so we're pretty excited for the conversation today. We've got two phenomenal scientists that we're planning on interviewing. Perfect. Paulina's already ahead of us. So folks, if you are coming in from different parts around the world, tell us where you are. That is always great to see. And so we've already got Paulina from Bulgaria. We got an exotic location right off the bat. So if tell us where you're coming in from.
So North American of you.
So North American. As opposed to, the various parts around the States.
Being a cultured European.
Yes. So yes, please let us know where you're coming in from. It's always great to see just how global the audience is here, and especially around the context of what we'll be talking about today, which is pretty cool. Which is just kind of this idea. All right, we think about clinical applications for these different cell therapies and the applications they could have inside of the clinic, but at the same time, right, we have this reality that not only do we need good clinical candidates. Oh, we got someone from Seattle. Perfect. But on top of that, we also want to have the next generation technologies that are going to make these cell therapies more effective, make them faster, and more efficient. Paulina, technically from Canada as well. So if you're from Bulgaria and you are Canadian, rock on. I like that, and Jeff doesn't.
I mean, I'm basically Canadian too. I'm from Buffalo, so it's like, what's the difference?
Fair enough.
A hop, skip, and a
Excellent.
jump.
All right. Well, continue to let us know where you're coming in from. But without further ado, let's bring in our two guests for today. And very kindly, they have added their last names in because they both have the same name from the same company. But Christian and Christian, welcome to the show.
No, thank you very much for having us.
And you're coming in from Germany. So instead of spending time with your families for supper, you're spending time with us. So we appreciate.
And I will say,
that.
I didn't even realize that your names were Christian. I was wondering before the show, Ben was like, Christian Stemberger, blah, 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 blah. And Christian. And I was like, why are you calling them by their, is it like a German thing to call them by their first and last name? But now I'm realizing that there's two Christians. Yeah, so when I said Christian, you're muted. You're probably like, which one?
Yeah.
All right. Well, then I guess I am the big dumb idiot that doesn't know anything.
Excellent. Well, I guess let's start with you, Christian Eckert. Can you tell us your role at TQ Therapeutics and a little bit about your background?
Yeah. So, my name is Christian Eckert. So I'm the CEO and one of the 4 founders of TQ Therapeutics. So my background is I'm biologist by training, biology, pharmacology. I started actually my career in the field of small molecules and antibodies, but then quite fast stepped into the cell therapy field. With the first company I've worked for here in Munich, we started the first clinical trial for TCR-T therapies in Germany. After that, I changed the company, so I went over to Juno Therapeutics. During that time, Juno Celgene, because Juno got already acquired by Celgene. That was the time when I met the other Christian. So they already have been with Juno when I joined. And there we were mainly focusing on process development, integrating new technologies, also for the Celgene portfolio for cell therapies, and then later on, because we got acquired by BMS, as you know, for BMS, in the end for the products which are on the market. So we were also part of that, and then after BMS, we started our own company because our vision, which we developed, seeing this whole development of the CAR-T field. So, we started in the old classic manufacturing field with this old classical manufacturing, and then we developed those technologies and those processes, and you've seen where it will go to. And with this vision that we have to take the therapy to the patient, to the bedside, we founded TQ Therapeutics, and we already are out there now for nearly 2.5 years. Yeah, so quite some time. So we built the company and the process around our core technology, our selection column. I think we can cover that a little bit later. And planning now our first clinical trial with our selection technology and our process.
Fantastic. Excellent. All right, and Christian Stemberger. There's a paper of yours that got shared recently by someone,
by a famous LinkedIn influencer, or so he thinks.
About molecular citrus. It was really cool. This idea, right, of how do you tune the response of CAR T once it's kind of done its job in inside of the body. So yes, please give us a bit more of an introduction of you, please.
Yeah, so another Christian and the third blue shirt, as I see here on the screen.
Yeah, Ben did not get the memo, obviously.
Yeah, that's
for having us.
Yeah.
So my name is Christian Stemberger, and I'm also one of the co-founders here of TQ and currently CSO of the company. And my background is in science, actually in immunology. And I have spent a fair part of my career so far in T-cell therapy, especially in regard to developing processes and processing technologies around how to handle T cells, how to streamline these processes in manufacturing, how to treat cells well, so to say, and also always with the focus on a little bit, at least, in bringing these therapies then also to the clinic and to the patients. And actually, initially, I started with non-ATMPs and only transitioned to gene-modified cells with the emergence of the CAR therapy, as Christian already said. So in the context of back then with Juno. Yeah, and from there we continued our thoughts and ideas and developments into what we've then also learned how the entire industry developed, right, and the struggles and the difficult parts about around manufacturing such a complex therapy in medicine. Yeah, and that's was kind of the founding idea of TQ.
So, I did a little bit of work before this to kind of figure out what you guys were doing, and here's like my take on the whole thing, and let me know how this lines up with your thinking. Basically, it's an autologous CAR T cell therapy. But it's not like most autologous CAR T cell therapies in that you guys do the manufacturing kind of at the bench or at the bedside. And you're able to significantly reduce the turnaround times because you can expand and select right there, and as opposed to shipping it off, doing the ex vivo, shipping it back, and all the cold chain and everything is a big mess. And the secret sauce is this cell selector you guys have gotten. And I have all kinds of maybe things you've probably thought about, but there's a lot of like biological advantages to this on top of the logistical advantages. So I'd be very curious to kind of know, like, what is your, how do you guys talk about it around the office sort of thing?
Yeah.
Excellent. Well, if I can interrupt you there, just one thing first, right? For anyone else who's coming in, we're interviewing two Christians from Germany who are focusing on bringing not only new clinical applications of CAR T cell therapies to the clinic, but also the technology to enable next generation therapies as well. So if you have any questions at any point throughout this show, it's always great to get your input and your perspectives, and we'll try to address them as quickly as we can throughout the show. And so by all means, if you've got any questions for either Christian Stemberger or Christian Eckert, by all means, feel free to put it in the comments below, and we'll get to it as quick as we can. So that being said, yes, sorry to cut you off there, Jeff, but I guess maybe if we just
I'm
start
used to it. It's okay. What else is new?
Yeah, fair enough. I guess, right, if we kind of come back to this central problem, like where we start this conversation, right? What is, like, when we look at the new technologies that we need inside of the clinic for CAR T, and we look at some of these unmet populations of cancer patients who could really benefit from these therapies, what would you say is kind of the main problem that you folks are looking to address?
So, I mean, as I said, we are coming from the classical way of manufacturing. So we've seen how long this process was, how exhausting it was. Is not just for the people who work on it also for the patients and for the cells. And I mean, many freeze thaw cycles, starting with an apheresis. We've seen people treated in Europe, and the apheresis was sent to the US and back to Europe, and things like that. And we've also seen clinical trials where patients really, it was not fast enough for them to get the therapy. It would have worked, but it was just not fast enough. And that was really something that we were thinking about, and said that can't be it. I mean, cell therapies are really, really good. So they really work. And it has to be faster. It has to be closer to the patient. And that's where we came from. So with TQ Therapeutics, we found that we have that technology which enables us to do this fast turnaround. So, and we have around this technology, around our selection chamber or the selection column, we developed several processes. The shortest is just a few hours. So it's no expansion. And it's just, we're using whole blood as starting material. So we'll just get about 200 mLs of whole blood from the patient. We directly run it over the column. And after, let's say, 30, 40 minutes, we have 100 million, 120 million, 150 million T cells with a purity 95, 96, 97%. Yeah. So, and with this, we start. And that is also, that's already something which reduces logistics, time, and everything for the patient. So that was the first point. And then what we do else is we don't expand. So we just use the cells as they come from the column and transduce them and give them back to the patient. So reinfuse them. So the whole process, our shortest process, I have to say, because we have a platform with which also can do other applications. But the shortest process is just a few hours, and we use non-activated cells. So because our column doesn't activate the cells, there are no residues on the surface of the cells afterwards. It can wash off everything. And with this, we try to overcome all those hurdles which cell therapy had during those times. And that was the problem where we came from.
Yeah.
That's a lot that I didn't even know. I have lots of questions. I'll let you go, though, Christian Stenberger.
No, I just wanted to reconfirm and put in shorter. So the entire idea was really to shorten the entire manufacturing process, bring it closer to the patient, and doing this by specific technical and biological treatments. But still having, the vision is to having ultra short processes. That's definitely our main driver of our work. But nevertheless, at the same time, maintaining a platform character. So also having the opportunity to extend here or there as individual cell therapies require, right? So there's different forms on how to genetically modify. You're having a big background also in lentiviral or in viral vectors, right? So there's different requirements and prerequisites for doing the one or the other technology, and still having the kind of this platform kind of way of thinking maintained to adjust and tweak if required, but still within doing as little manipulation as possible with as fast and as cost-effective turnaround times as possible.
So I'll start with this very low-hanging fruit question. Is it just T cells, or can you do NK and macrophage and neutrophil? Can you do different selections, or is it just T cells?
So with the current column, we are focusing on T cells. There's a specific way how we catch those cells. It's via FABs, which are on the surface of a matrix, but we easily can expand them to other cell types. So you name the cell type. The more specific, of course, the surface marker is you target with the FAB, the more selective is the column. And that is easy done in the end.
Right.
Yeah.
So, okay. Well, I won't get into the weeds just yet, but there are a lot of myeloid-based cell therapy platforms that might benefit from this significantly. one of the issues with myeloid cells is they just don't live long enough to do anything. And if you can get these things extracted and get them selected in the hospital, yeah, you might be unlocking a completely different type of like an immune cell based platform therapeutic. So, okay, well, that's the first question. The real big question is, you're not expanding these things, right? And I'm going to guess it's enough for a, okay. Okay, so what is the typical dose? Is it the same as a standard CAR T cell, or is it? Can you guys do multiple? Can you create it up for multiple doses? Do you not care about that? You just kind of infuse what you got.
Well, this depends on the starting material actually, and of our unique selling points. I would say is that we, and this is also where we focus on, is that we try to not start with the standard leukapheresis materials as the standard. So we could and we can. The system is also capable from a size perspective to handle these kind of starting materials, but we do aim at whole blood selection. So just to again with this idea of again bringing it closer to the patient and making it more nice for the patient. So we do start with the whole blood, as I told you, and as a rule of thumb, so from 200 mils of whole blood, you would typically get 1.5 times 10 to the 8, so 150 million cells selected, T cells selected, that you then take into the process. And of course, depending on the transduction modification efficacies and so on and so forth, you do get quite significant transduction efficiency and ultimately then a good dose. That's number one. The second one is, since the overall process is fairly short, there is the option for a redosing scheme, right? So you could just a week or two later can do another one, right? If you would, for instance, go for leukapheresis, then the limit is way higher. So then we're talking about billions of cells that can be handled and processed at a time.
And you guys do the genetic manipulation right there as well? It all happens kind of in the same closed system?
Yeah, it's altogether it's a closed system or nearly a closed system. It depends, of course, what kind of mechanism you use. I mean, we can use, so the system is completely modular. So you can use virus, you can use LMPs, you can use electroporation, you can use mechanoporation, whatever you want, we can do it with that column. I mean, and that also, of course, depending the length of the process is also depending on the method you use. I mean, a viral transduction, of course, will not in 4 hours. It will be a little bit longer because you have to activate the cell. Yeah. But yeah, so we can do all together. Yeah.
So Kathy just put a good question here, and I was going to ask this next. I have a huge question around QA, like the quality assurance of this and like the actual like consistency between batches. I get that there's all kinds of inconsistencies with regular autologous, but the quality assurance is a little bit more robust, with a traditional ex vivo manufacturing process. How do you guys do this? I'm going to guess you're going to have like specialized trained physicians or people in the hospital by hospital. Do you guys have? I'm guessing you already have kind of like logistical plans put in place to get people, able to do this consistently across the world, hopefully, right?
Yeah, absolutely. Yeah. So, but this question is an excellent question, and there was also a question. Of course, we were thinking about a lot. So we were talking also to the regulators, to European regulators, for example, to the German authorities, to the Swedish authorities about that. Because I mean, in the end, if you have a 4-hour process and you have to do release assays, which needs 2 days, then the whole process
what's the point?
Absurd. I mean, what's the point then? Yeah. So, and there was a little bit the point for us. So it was, what do the regulators say? So we went to the German regulators, to the Swedish regulators, and they basically said the same. So they said there is some kind of testings we have to do in line, so in the process, but those testings are in line with the time we have. So it's like viability, purity, things like that. You can test quite fast. Yeah, and other testings which you usually have to do, like CAR positive cells, CD3 positive cells, things like that, we can do retrospectively. Yeah. So that is the plan for the release. Yes.
I mean, as we call our idea hybrid in vivo approach, right? Because it's so fast that the actual biological functions starts to work out once the cells are already back in the patient, in the circulation. But you still have this kind of manufacturing part upfront where you can highly selectively isolate your cells and modify them with a high degree of control, obviously, before you infuse them back. Also, very little residuals going back into that. So as this is a hybrid approach, so to say, also the analytics is kind of a hybrid approach. And that's exactly what we've already been from the early stages on, we've been discussing and involving the regulators, because that's a very important question, as you can imagine. So there is a little bit of a different quality control kind of procedure around that. Being as Christian said, so there is built into the process analytics, just to make sure you're transferring cells basically to the patient, and what number of cells actually, and how viable they are. But all the strengths, potency, and safety and whatnot assays, because of that fast turnaround time, they are put a little bit to the back burner and are analyzed retrospectively, right?
Standard FACS analysis, for example. Yeah.
Sure.
Nice.
Yeah.
So, I guess that also helps address Brian's question that's in here as well with regards to the short 2-hour process. A lot of these different analytics and QC releases can be done retrospectively, which is awesome. And that's a huge critique that people have had so far, right? It's like, okay, you've got this therapy that's now waiting in a freezer somewhere while the patient gets increasingly sicker. It'd be better for them to just get it now and then do some of the analytics after. So that's awesome to hear.
Yeah.
Okay, so just I guess maybe to recap here a little bit, right? For people who may have not come in since the beginning of the conversation, we're talking with TQ Therapeutics, and they have developed both a really cool technology for sorting T cells out of whole blood samples that allows you to do very rapid CAR T production. And they believe in their technology so much that they're not waiting for someone else to do it. They're also developing their own clinical assets to take into the clinic. So I guess we've been talking about this so far, but we haven't actually seen what this cell sorter looks like. I understand that you guys have one with you. Would you be able to kind of show us what it looks like and how it works? Hey.
Ooh. And the crowd goes wild.
With this powder in there. The nice thing about that column is that it's fully sterilizable, so you can take it off the shelf. It's stable, and you just flush it once with PBS, and then you put the whole blood on it, and that's it. Yeah. So it is easy to use. Yeah. It's a very nice system. Yeah, I have to say.
So
that's the whole thing, huh? That's the secret sauce right there.
That's the secret sauce.
That thing might revolutionize CAR T cell therapy. Look at that.
Let's hope.
So I guess just to take me through that. So you've got a column that's probably like 2 inches tall, maybe like 1.5 inches tall. So for the rest of the world, that's 4 centimeters.
For those of you on the wrong system.
So what's the capacity for this little column? Like how many cells can that collect?
So from whole blood, of course, it's 150 millions, because in 200 mL of whole blood, what we use, there is not that much more. Right. But from apheresis, if you use apheresis, leukapheresis, you can catch more than 2 billion with this column here. Yeah, for that size.
So is it a negative selection, or is it a positive selection? This is probably superfluous.
That's a positive selection.
Positive selection.
And, the way it works is from the protein field, it's basically an affinity-based chromatography. So we have the resin that is functionalized with sandwich functionalized with several proteins. One of them being the actual FAB ligand, so a part of the antibody that then actually interacts with your target cells. And you had the question before. Yes, it's modular, because you can with Lego bricks, you can exchange that ligand to another specificity.
Yeah.
And once you put this column to the buffer, since everything is pre-confectioned, you can run your cells over that, and you positively select out your target cells because they get retained. They bind to the surface of the resin. They get retained, and the unwanted cells are washed away. And ultimately, you can then elute with chemically, so with biotin addition, very gently, you can break open the complex and elute the cells from the surface. And on top of that, the actual ligand were engineered in that regard that they have a very fast off rate. So once they are monomerized, spontaneously dissociate off the cells.
Yep.
And that's also the reason why we do not introduce any biological signals to the cells unless wanted.
Yeah.
But under controlled conditions, that's a very gentle way to select the cells. And secondly, you also reduce almost all of the residual reagents that you don't want to have.
So, pardon my naivety, but this is what enabled this technology to happen now? Why hasn't anybody done this in the past? Was it just a good idea that you had, or was it something that happened biochemically that you guys discovered? What is the actual, I guess, the secret sauce to the secret sauce? If you can get into any kind of I don't want to get proprietary, but
I think you can.
Yeah, well, I think the way it was developed, this comes from a long-term development. Obviously, there's a twin technology, so to say, on that is aiming at cellular activation. So that's an activation reagent, still with the same biochemical and ultimately cellular properties, so that you have a super tight control on what you're doing. So that's basically the idea and the driver of developing that. And we came forward in combining that, obviously, right? Because what you can do is you can also combine several unit operations basically into that reaction chamber if you want. So you can not only select cells and retain them on the column, but you can also manipulate them once they are still immobilized. Like with genetic modifications, for instance, or with activation, whatever the process looks like.
You can do the gene editing while they're in the column.
Yes, you can do that.
Yeah, that makes sense.
That is possible. Yeah.
That's really cool. So then, what about the possibility of doing sequential selections?
Yeah, also possible.
Yeah. To get
they're letting on more than they're able to.
Yeah, that's actually this is very, very cool technology.
Yeah.
And one of the questions that I had, my dumb brain was just what's the difference between this and just doing allogeneic? Because the idea is let's save some time, and allogeneic is, it promises to be off the shelf. Then what is the point of doing the quicker autologous when we already have allogeneic platforms available? So what is your thoughts on that? I have some thoughts there, but I'll let you guys answer for yourselves.
So the huge advantage of allogenic cell therapies was, of course, the off-the-shelf idea behind that. For that, unfortunately, you have some downsides. So you have to really manipulate the cells extremely. Yeah, and that is, I don't know how good that is for the cell. And it's a viable therapy. Please don't misunderstand me, but that's the thoughts we have around that. Plus, I think the batch sizes are not that big as they thought in the beginning, perhaps. So it is also some effort to get those batches done with all the regulations around it. And why using an allogenic therapy off the shelf when you can use an autologous therapy off the shelf? So
that fair. Sure.
Yeah, but iPSCs is valuable. It's good therapy. So please don't misunderstand me. But it's of course we are more on the autologous side.
Right.
Yeah, well, I think the big advantage of autologous cells is simply that they require way less of engineering. Ultimately, at the end of the day, they are immunologically perfectly matched, obviously, since they are autologous, right? And the two things you can do is in scaling is either you scale up your entire manufacturing, right? But then the scaling has to match kind of your output parameters. So does it really scale big enough, so to say, to really have a good off-the-shelf product?
Yeah.
And the other thing is to use what you have, and that's autologous cells, plus the added benefit of having certain biological benefits, I would say.
And I mean, also one of the ideas of TQ Therapeutics was, of course, to manipulate the cells as less as possible, and so that is also possible with our column.
Yeah. So I want to get your opinion on this biological phenomena. People have noticed that when allogeneic CAR T cell therapies began to be tested, one of the big issues obviously is rejection. And people go through, they jump through 15,000 hoops to make these things stealth, and they still see relatively low persistence. So much so that it's affecting efficacy, and and I think that the field kind of thought, okay, just make them more stealth and make them more defensive. We had a company on maybe 2 months ago now, called Genicity. And they're doing a thing where it's an allogeneic cell therapy, CAR T cell therapy, but instead of deleting, and also the problem with allogeneic obviously is graft versus host, right? So that's one more genetic manipulation. Let's delete the TCR alpha, get that thing out of there. No more graft versus host, theoretically. People have realized that one of the problems with allogeneic CAR T cell therapies, allogeneic T cell therapies in general, is T cells really like their TCR, even if they're not using it. And there's an idea, this is a biological concept that's maybe not proven fully yet, but there's an idea that simply deleting the TCR from the T cells makes them sad. And they might not be able to persist as long because they need their TCR, even if it's not providing them like actual TCR MHC signaling. One of the problems with allogeneic is that what do? You need to delete the TCR, otherwise you're going to kill the patient. This company had some genetic modifications to a TCR to kind of get around that, but I think your approach is better because you don't have to worry about deleting the TCR. They're autologous. And you don't got to worry about doing stealthing because they're autologous. So it's kind of like getting the best of both worlds.
It's the same as we always say. I mean, of course, everyone knows now. Everyone or a lot of people are going in vivo. So in vivo therapies. It's. I think for us,
don't
in vivo.
I'll join the pitchfork mob against in vivo, especially in oncology.
But that's exactly the thing, I think. So I mean, all those therapies have their right to exist, and they will exist in parallel. So they will all go their way, and they will find their niche, and that is the important thing around it. And I mean, in vivo is, of course, it's also in autologous now. You have those two main streams. The one is clearly the manufacturing. I mean, manufacturing, but in a fast way, like one-day processes, for example. And then you have the in vivo path. So, and I think that is, in my opinion, that is a good way to go. And with our technology, we can also bridge those two technologies quite well. So, I mean, that's the hybrid in vivo. So we are not really in vivo because we have this whole step outside the body, but we are nearly as fast as in vivo because after a few hours, we're back in the patient. So we are competitive cost-wise with in vivo. We are, but we also have the safety and everything around the classical autologous system. So I think that is for us the more important way. So allogenic is definitely also has its niche, but I think we are completely focused there, of course, on autologous.
Yeah, I think there's so many parameters that have to be put into the equation, right? On deciding really one or the other. But here and today, that's maybe more a kind of a discussion for, let's say, in 10 years or so. But
I agree.
For today, because you were mentioning making them more stealthy and modifying here and there, I mean, that's all super approaches and also working, right? But today, I think every additional manipulation step simply is just from a practical perspective, it takes time and costs money. And the pressure cellular therapy currently has is exactly time and money, I would say. And So I think I'm really excited on how the field will develop in all of these aspects, right? But for today, I think that's again one for us an important aspect to keep things simple and to have a fast process, right?
So speaking of keeping it simple, does your protocol involve any lymphodepletion preconditioning?
No.
Excellent.
There you go, Kathy. There's the answer to your
Oh,
question.
there you go. Okay. I see. I put it to full screen to look at the device, and I'm not seeing the comments now. I did not ask that because Kathy asked that. I would have pulled the comment up. No, but that's very, very, very good. I mean, I feel like I've seen people are really moving. When I first heard of some companies who were like, our approach requires no lymphodepletion, and no one cared. 2 years ago, no one cared. And I think that I haven't seen people agree that lymphodepletion is a problem, but I've seen more companies who are focusing on eliminating it, which means that I think it's kind of happening, in the shadows. People are realizing, okay, if we can eliminate lymphodepletion, it really opens up a lot of opportunities. Especially, I think that the elimination of lymphodepletion by itself opens up, I think, 2 really nice opportunities. Let me know what you think about this. The first one is elderly patients or heavily pre-treated people who wouldn't normally qualify for a preconditioning regimen. That's the first. So elderly people and people who are heavily pre-treated. But then the other one, I think, is autoimmune disorders. Because with oncology, I think that you can justify a pretty intense therapeutic regimen to kill my cancer, because if you don't do that, my cancer might kill me. With some autoimmune disorders, barring lupus nephritis and things like that, typically these patients aren't facing a lethal issue. It isn't like a life or death thing. So having this crazy 6-week lymphodepletion regimen might be like, you know what, I don't really want to do that. But getting rid of that kind of opens up that potential, and it reduces that barrier to entry. What do you guys? That's probably relatively well known.
Yep, but it's absolutely right. So, I mean, we were talking a lot to our advisor. We have physicians as advisors, of course, who also advise us in regards of the clinical trial we want to have, and especially in some autoimmune diseases like MS or something like that, they're desperately looking for therapies which do not need a lymphodepletion. Yeah. So, and that is exactly the way we go.
Yeah.
So no, I mean, of course, I mean, if we get the blood from the patient and put it on the column, it's not very useful to do a lymphodepletion first. So.
True. That's true. Yeah.
That's so for us, but it's the same for in vivo. I mean, doing a lymphodepletion and then an in vivo therapy is the same. It wouldn't work. So, I think that is the way to go. Definitely. Yeah.
So then, sorry, Ben, I'm just going to hog this. So
we should get to some questions at some point.
Okay. One more, I promise. This is the last one. And then I'll shut up. Because you guys don't do any kind of, or at least your primary process, as I understand it, doesn't involve any kind of expansion or manipulation molecularly besides the genetic manipulation. Are you worried? Now I understand that you're also not working in oncology right now, but are you worried that potential, the starting material will just be so inconsistent, and you might have patients who just don't have, their T cells are all exhausted, for lack of a better term, where you're not going to be able, no matter, because one of the advantages of ex vivo is we can kind of like ensure that we're going to get a nice juiced up population of T cells. With this, if you're not doing that, you might risk, okay, they're all CAR T cells, but they're kind of wimpy.
Yeah, that's true. I mean, of course, that's always some sort of autologous treatments, right? This is always a potential threat. Nevertheless, I think there's even quite some data out there that, again, depending on the indication and so on and so forth, I think you can pre-screen, obviously, right? And secondly, there is emerging evidence that in certain indications, particularly in these autoimmune indications, the cell products that have been published and reported, the manufacturability was quite good and was not really seen as a big threat to kind of have these shortened processes around.
Right.
Yeah.
So I guess just one more little comment. I guess the way that I'm reading it then is it's kind of you're I don't want to overstate this, but I feel like this is like just superior to allogeneic and autologous. But if you're going to go the allogeneic route, it's like this just seems to kind of solve the problems. I feel like most of the allogeneic field is like focusing on the genetic manipulations. All these big allo CAR T companies are like, we have a super fast way of doing genetic manipulation, and our secret sauce is our genetic editing technology. Whereas you're saying we don't really need to do any of that. We can just use the autologous and do it quick enough to kind of get rid of all those problems.
Absolutely, exactly. So that's the time is the magic here, and with this technology we are so fast, so it is off the shelf in the end. I mean, you could also use that and do manufacturing in the classical way if you want to do that. It's not a problem with that technology. But if you want to go fast, you're so fast, it doesn't matter. It's off the shelf in the end. And if you don't have to manipulate the cells, and if you use the cells as they come from the patient, I mean, they come from the patient and they go back after a few hours into the patient. So there's even no CAR on the surface when they go back. So that all develops in the patient as for an in vivo therapy. But and there is also nothing done to the cells. They're not activated at all. So we tested that. We looked at that. So there's no activation. There is no phenotypic changes after selection, nothing. So that is the pure cell as it came from the patient, goes back into the patient with a little addition of a mRNA, for example.
Yeah.
Phenomenal.
Well, you got me sold, guys. I like it.
Where does Jeff sign? Yeah. Okay.
Can I come
so
work for you? No.
All right. So I appreciate everybody who has very patiently waited. There's a backlog of questions here. I'm going in no.
You can just blame Jeff. It's fine.
We won't go in any particular order. Here's a question from Shashwati. Are you able to select T cell subsets?
So in principle, yes. It depends, as I said, on the surface marker, what you want to select. We could easily make FUBs against specific surface markers, and then we can select also cells CD29, CD27, 57, whatever you think about. Yeah. We also have a matrix for CD4, CD8, for example, that's already in-house. Yeah, so it's definitely doable.
Yeah,
gotcha.
There it is.
Does that kind of lead into this next question then?
Yeah.
Currently we don't, but if you want to say something.
Yeah, so I mean, yes, we can. That's the basic idea of the entire modular system. To be honest with you, for the moment being, because we obviously want to develop also a therapy and bring it to the clinic. So we do focus on pan T cells. So CD3, that's our go-to marker that we're currently using. But we have developed also markers for subsets, either the big ones like 4-8, or as you already mentioned, even more in further down the
line
resolution, so to say, in subsets. And what we've also already done, again, this is not something that we are currently pushing to the max, but we have proof of concept on that, is doing serial selections, for example, right? So then you first do a CD8 selection and then go into further down into like selecting more less differentiated cells, for instance, with L-selectin or.
Fantastic.
Which is a huge issue with ex vivo. I've read so two things. There's plenty of literature to suggest that getting a relatively even ratio of CD4s and CD8s actually helps CAR T cells. This myth of CD8s are the golden standard. It's maybe with TCR T cells, but even that, like I think that getting both the helpers and the CTLs is crucial so doing pan T is the way to go.
Absolutely.
But then number two, I think that part of the problem with the ex vivo expansion is that you get a lot of CCR7 expression. I've read a couple of like big reviews that have showed that one of the issues with ex vivo expansion is they're getting like lymphoid T cells. And the actual subset's escaping my mind, but you're getting cells that kind of go to lymphatic tissue, which is great for lymphoma. It's great for leukemia. It's not so great for solid tumors or for other kinds of indications. So I think that, with your thing where it's like, hey, we're just going to grab these cells and just get what we got, do a pan T cell selection and pump them back in 3 or 4 hours, you might be actually getting that value as well. That's all theoretical, but we're not going to be creating bias for some kind of subset.
Yep.
Correct. Yeah.
And
so you definitely observe in extended expansion cultures and so forth. You do definitely observe kind of a curtailing, like a filter effect, so to say. So you lose the more, let's say, at least phenotypically, but also then translates to function. You lose more the effector late stage differentiated cells, but also you lose the earlier front, so to say, and get out this middle section, so to say, with all the effects you just mentioned. Correct. Yeah.
Very good. Okay, so we've got a bunch more questions here. One more from Kathy. Kathy's been putting a bunch of banger questions in here. You mentioned you can electroporate. Can you integrate into the MaxSite system or other and perform additional modifications?
Yes, in principle, yes. We're relying on a different platform for the moment. Yeah. But the entire system is a closed system, obviously, right? And we can also control this on an established platform from our collaborators. And we can also go back and forth with these cells and include in washing and buffer exchange steps, so to say, for electroporation. But the MaxSite system would be possible, yes.
Gotcha. Yep. Okay, here's some really old questions, and by old I mean people who were faithful and asked questions early and we skipped over them. Here's one from Paulina. What is your perspective on the translation of cell-free therapies? How do they compare, for instance, exosomes? So I know we've had a little bit of a conversation about kind of this approach of doing autologous in just a few hours and then putting it back into the patient versus in vivo, but maybe if you could just summarize in case anyone missed that part of our discussion from earlier. Or I'll leave it up. Here you go, so you can see it.
No.
So, like, sorry, maybe a better way to frame this would just be, can you kind of compare, contrast this approach versus in vivo therapies going after the same thing?
So the question is if we can compare our technology with the current in vivo therapies. Yeah. So how do I frame that? The effect or the result will be probably the same. Yeah. So there will not be much difference. I think the difference comes from the starting of the process. So if you use, exosomes, LNPs, whatever, and you have to produce or have to put much effort into the beginning of the manufacturing, where we can easily use everything which is there without any modifications. But for us, of course, we need an application system, which we have, and where our effort comes into. But I think in the end, in the patient, the result will be probably pretty
better.
Similar.
It'll be better. I've got it out for in vivo. I got it out for in vivo. I think that I'm becoming a little bit more of a believer, maybe a little bit, but I've had it out for in vivo since day one.
It's good and it's hot and it's good out there. But let's see in vivo in regards of indications, in regards of integration, stable versus transient, and things like that, and regulations around the whole topic of in vivo. It's there are so many good scientific puzzle pieces out there, but getting them into the patient in the end and make them commercially and clinically viable is always a different story. Let's see. Next year we're probably we will know much more. After that year. Yeah.
Sweet. Okay, so for a good chunk of this show, right, we've kind of talked about this technology and the promise that it could have. But there is one question here, just with regards a bit more to the clinical side of this whole thing, which you guys have this really cool technology, makes CAR T ideally going to be a lot faster, autologous CAR T therapies. But then the question here is just what are your target conditions, and are any of your products in clinical trials? So I guess just like, can you talk to me a little bit more about what your clinical trial plans are for kind of the first shot of this?
So it is not yet been to the clinic. It is ready for the clinic because we already had that at the brink to the clinic. Yeah?
Right.
So we are planning the clinic will be an basket trial in autoimmune disease. Yeah. So I don't want to go into the indications but it's a basket trial. So we have several indications, and I mean there is not. I mean we won't be very special in regards to others, but I mean we are planning to start that clinical trial as soon as possible. So probably end of 27, beginning 28. Yeah. It also depends on the manufacturer of the GMP columns, because it's a lot of biologics, so it's expensive and it's time consuming. We gladly have a very good partner with doing that with Fresenius. They in-licensed the technology. So then we're gonna, as I said, do that clinical trial, that basket trial, in autoimmune disease. But it's not that we also can do, of course, stable integration, but for us, we want to prove the technology, the proof of concept, and that is done in the clinic. And because I mean, we're a small company, so we don't have a lot of shots on the target. So we have to have the first one has to hit, and that is the easiest to go currently. But we also plan for other indications, like in oncological settings with stable integration, but that is on a longer timeline. So it's not in the next 2 years. Yeah.
Very good.
I'm still dreaming about the myeloid cell therapies over here. I think that sounds really, really interesting. If you're able to, I mean, like, because there's way more neutrophils than there are T cells, right? I mean, like, you can get so much, you can get so many of those things. What's that?
Yeah, absolutely. Let's have a meeting about that sometimes.
Let's do it. I got a few companies who might need your help. Yeah.
And they're quite sensitive cells, right? So having a gentle methods in place to utilize them is key.
Yeah.
Fantastic. Okay, there is another question here. So I guess this question's coming in from Richard Jones. Richard's also been has had some great questions over the last couple of months on Bio to Bedside. Thanks, Richard, for coming in. So the report last week about giving checkpoints before 11:30 a.m. gives statistical difference in survival. So taking T cells at 7 a.m. may be better than 2 p.m.
Something as a immunologist.
Some circadian rhythm stuff. Dang, I like it.
But like maybe hopefully this is tangentially related to your comment, Richard. But the one thing that I also am very interested by this concept, right? Normally for an autologous therapy, right, where it's your own cells, they're going to take these cells away. It's going to take several days for you to kind of get them processed, and then through the entire process, then back into you. But I would be interested if you guys have, but one of the things that's really interesting with some of these other people who have done like fast CAR T, when they're like talking about like 24 hours, and you guys are even shorter than this, is that actually with these shorter term incubations and like lead times, like time it takes to make the therapy and put it back into the patient, you actually end up preserving stemness of the T cells. And by preserving stemness of the T cells, you actually make this therapy have a much longer durability inside of the patient, just because you've got this additional ability to create long-lasting memory. I'm curious if you guys have seen anything with that so far.
We did. Of course, again, to be fair, I mean, this is more of a topic in a let's say hemato-oncologic setting or a tumor setting, right, where you really need persistent cells. Not so much in where we are aiming for our lead, so in autoimmune, where actually the contrary is more or less true. So you want to have a self-limiting system, because these patients are not mostly not kind of with the back to the wall,
right?
Right, and you want to have a fade out period, so after you achieve reset, getting the B cell compartment back. But back to the question, we've observed this in earlier research we've done and processes where we looked at the cells, that you do see exactly that you preserve stemness, if you want to call it like that, but you definitely generate cells that are highly proliferative in vivo. They are very well maintained. They can self-renew, and by this, ultimately also that translates into the option to reduce the actual cell numbers that you have to transfer in order to see the same clinical effects.
Mm-hmm.
And that has been not only by us, but by many people out there, in groups out there, has been shown that this is the case, actually.
Yeah. And the preservation of stem is likely an artifact of the expansion process, I would think. I mean, the more and more you expand, you're just going to dilute out any cell that's not going to be activated.
Drive the cells in a specific direction with the expansion.
Yeah. So, but I mean, with no expansion, a quick turnaround like that, you're basically taking over allogeneic. And I think,
Hopefully.
Yeah. I mean, like I said, I've been watching a lot of the allogeneic companies kind of scramble, and a lot of it is just like, we need better genetic editing, quicker, more accurate, and we can do more edits at one time to get we have to have 15 different things in there to make sure they're not going to kill the patient or die, right? So
and it's great. Obviously, it's an effort and it's expensive.
It's expensive,
right?
And it's
yeah.
Right on.
Hopefully we can overcome those issues.
Yes. Excellent. So I guess for anybody who is currently developing a therapy or manufacturing people, what's a way that people can partner with you guys?
The best way to contact us is via our homepage. We have info@TQ Therapeutics email address, so just write us, or via LinkedIn. So we are all active in LinkedIn. Just write us a mail or via LinkedIn, and we will come back to you. Definitely. Yeah.
Perfect.
And our colleague will be next week in San Diego.
So,
Christine Zündorf.
Yes.
We'll be there too.
We'll all be there.
We will be there together with Fresenius at the Fresenius booth. So if there are any questions, any way to connect with her, feel free to. She's happy to talk to other people. Yeah.
Excellent. So, folks, if you're at ATW next week, you can stop by the Fresenius booth and you can chat with. It's Kristen, right?
Christine, of course.
Christine's going to be at the booth, so you can go chat with her about TQ Therapeutics. Awesome. Okay, actually, right here at the 11th hour, we've got another question from Kathy. Kathy's just holding down the fort over here with some amazing questions. Can you comment on the reduction of COGS and cost of therapy compared to traditional autologous therapies? Ooh, good question. How have we not talked about this yet?
Excellent question.
Maybe I dreamed that.
In the end, we are currently calculating that. What we can say now is, our prices, of course, the COGS is, of course, much lower than the classical. So it's not comparable with the classical manufacturing. It will be very, very competitive with in vivo. Yeah, so we have actually some nice advantages to in vivo, that we can exactly calculate the amount of vector we need, for example. We don't overdose, nothing. So it will be competitive to in vivo. So it's the exact numbers we can't say, but it's very competitive. Yeah.
Mm-hmm. I believe it. Yeah, and I guess the other thing too that we haven't talked about too, right, is hopefully this doesn't sound like Jeff and Ben are just doing a sales pitch for you. But the other thing too, right, is there's not really any cold chain at all.
No, not at all.
Right, and that's just an entire very expensive component of it. It's completely gone.
There's a little bit of a cold chain in regard to the genetic material.
Right. Yeah.
Right. You have to store that at a point in time.
Yep.
But it's a completely different logistical burden, like the current manufacturing efforts that you have with handling cells and across continents and several free store cycles and you name it. So it's massively easier than before.
Yeah.
Nice.
So no freeze thaw cycles for the cells itself.
That's the biggest thing. Yeah, the freeze thaw for the cells is the biggest thing for me. Yeah.
Phenomenal. Well, gentlemen, we're right at almost the end of the hour here, and it is much later where you are compared to Jeff and Ben, who are just chilling out around lunchtime. So we will let you get back to your families and your regular lives. But thank you so much for coming on. This was great. Folks, if you want to learn more about their technology, go check out their website. There's an email address on there that you can reach out to. Thank you again to both of the Christians for coming on, and we look forward to watching what you guys do next. This is super cool to see a company that's taking
very
both technology and clinics to the clinic. So right on.
Thank you very much. Yeah.
Thank you guys. Cheers.
Nice day.
Take care. Bye bye.
Stick around for 1 second, guys.
Yeah.